Hi, everyone. Welcome to the Live Lounge here at London Calling 2024. My name is Rachel, and I am a technical product manager here at ONT. And today I am excited to talk to you about the capabilities of PromethION and why it is such a great tool for unlocking new potentials in human genomics. However, the true highlight of our session will be hearing from Dr. Carika Weldon, who most of you heard from this morning, and she'll be giving kind of a behind-the-scenes look about how she got her lab set up and how she's using PromethION. But before we get there, first I want to talk a little bit about what you can do with PromethION. So with a single PromethION flow cell, you can get 100 to 200 gigabases of native gDNA with a N50 of over 25 kilobases. You can get over 100 million reads for cDNA transcript analysis. And you can also get methylation data, direct RNA sequencing, reads from short to ultra long, and much, much more. One great thing about PromethION and the PromethION flow cell is that it's the same across all platforms. So no matter if you are working with the P2 solo, P2 integrated, or all the way up to the P24 or P48, you can be assured that you're getting the same amount of data and the same quality the whole time. This capacity and capability explain why the PromethION is so great for human genomics and genomics research in general. Because we have such a different range of devices from 2 positions in the P2 solo and integrated all the way to the P24 and 48, you can be assured that there is a PromethION that scales for every experiment. However, today we're going to be focusing on the P24. So with the P24, you can get 48 30x whole coverage genomes per week and about 2,500 genomes per year. But as you can imagine, that obviously scales way more when you add more PromethION P24s into your lab. For example, if you wanted a whole fleet of them. However, with more P24s comes more data. And I'm happy to say that now it's easier than ever to set up data analysis on EPI2ME with your PromethION. So in the next upcoming MinKNOW version, we are going to have an integrated setup for the EPI2ME human variation workflow while you're setting up your sequencing run. So we've taken the most common and most important settings from the EPI2ME workflow that you know and love, and I've put it into MinKNOW. And this integration ensures that the data is always going to the correct location. You don't have to think about where to send it. You don't have to think about setting up the analysis after the fact. When you set it up in MinKNOW, it queues it to run as soon as sequencing finishes, so you can be assured that you have the shortest time from sequencing to answer. Additionally, you'll see the status of the workflow in MinKNOW as well. So you'll see that it's queued, running, or completed. And when it's completed, you'll see the EPI2ME workflow report available in MinKNOW as well. And this report gives a lot of different information. It's a very nice interactive format. And based on what you select in your settings, you'll get information like SNPs, SVs, and much, much more. And we're also happy to announce that if you're looking to do further analysis with the human variation workflow, in EPI2ME itself, we've integrated a few of our tertiary analysis partners in order to make it even more seamless to get even more data out. So currently our partners are GenX, Fabric Genomics, Varsome, and Seq1. And after running the human variation workflow in EPI2ME, you can now, if you buy credits through your Oxford Nanopore account, go directly to one of these partners, so you can go even further with your human genome analysis without having to think twice. So basically, all these new advancements in analysis, including EPI2ME and MinKNOW and tertiary integration, are just a way to get your data out even faster and get results faster than you could before when you had to think about different parts of software and different applications. So I could talk to you all day about the power of PromethION, but I think it's better if you hear it directly from one of our amazing customers. So I am really happy to introduce Dr. Carika Weldon. And Dr. Carika Weldon is the founder of CariGenetics, whose goal is to study genetics of Caribbean people to learn more about their specific susceptibility of disease, genetic markers, and also to add more general knowledge about this population to research. So without further ado, Dr. Carika Weldon.
Thank you so much, Rachel. I'm so excited to give you now the behind the scenes of exactly how it all went, because it was not smooth sailing. We addressed this already, but just in case anyone missed it, Bermuda is a real place. I invite you all to come. It's only a 6-hour flight from London to Bermuda. I see eyes going up, yeah. And it's sunny. Oh, it's sunny outside now. I think I managed to bring some sun from Bermuda. So prior to doing all of this, in terms of setting up a lab in Bermuda, my experience was mainly on MinKNOW. I actually taught myself how to do it. I happened to find it when I was in my own lab after my PhD, because I wanted to look at modified guanines, and I found that you could do that with MinKNOW. And I happened to then end up at the Oxford Genomics Centre. I had a little bit of P48 experience, but that was it. So here we go. So again, as I mentioned earlier, a year ago, we just about got ethics, and we had just about secured some funding as well. And the idea was now able to become a reality. It was easy to order online, so that's one thing I want to address. Oh my goodness. So I know, like, I was in England for 12 years, and you're working at, you guys, in the UK or the US, you can go and order today and get it tomorrow. How many people have experienced that? Yeah, I mean, it's standard. Like, you're like, oh, I need a new ligase. Let me just order it, and then it gets there the next day. Not the case in the Caribbean. So it takes weeks to get things, and for some companies, they actually make you go through distributors. And because LATAM and the Caribbean is usually grouped together, we tend to have to do with Spanish-speaking distributors, and I don't speak Spanish. So it becomes very cumbersome. And so being able to just go on the Nanopore website, order, like, I'm going to buy some shoes, actually I found to be really, really helpful. In some instances, we needed more reagents, and I was able to do that late at night when I realized we needed them, and then they were shipped within 2 days. So I think, for me personally, it was very easy to order with Nanopore. Okay, so what I didn't say earlier is we had ordered the PromethION. It arrived in Bermuda, but the first hurdle was our government got hacked. Who would have thought, right? This is last September. And so what meant was we couldn't get the actual PromethION out of customs because the government had to process the customs form, and their whole entire system was down. And we were trying to figure out, like, when are we going to get it? And so we actually had the FAST team arrive without the PromethION in the lab and train the lab team without the PromethION. Like, what are we doing, right? We did have a MinKNOW. We had the MK1C, so they were able to kind of learn about loading flow cells, but it's very different for a PromethION and a MinKNOW. So that was the first hurdle. Luckily, once we did get it, it was very easy to plug and play. We actually also had to change a few electrical things, but we managed to do that. We set it up before December. We did try to do the certification, but the shearing version didn't quite work. We didn't get that right. So we packed up for Christmas, and then we came back in the new year. So, as I mentioned, on June 8th, we came right in on June 2nd and was like, let's get this done. We were able to get certified on the second try. And as you see, I actually took a screenshot. I was so excited. I actually went on the website before and took a screenshot of the map, because it has like a map with all of the certified locations. And I took it afterwards so you can see the before and after. Very dramatic. I was like, let me just show, we're putting Bermuda on the map. Right? All right. So we are the first in the region to get certified for PromethION. There are 2 others in Latin America for GridION. And with that, we went straight into the 102 breast cancer samples. So we are a small startup company. Luckily, we have a partnership with our local college in Bermuda. And this is our setup, very humble beginnings. This is the lab team. And I have to shout out Natalia, who is our account manager. So she's in that picture there. And we found for us, we wanted to get this done. We initially wanted to get it done by end of January, but we saw that things were taking a little longer, but we wanted to make sure it was sustainable. And so we basically had one of each of the lab team doing DNA extractions, one doing library prep, and one loading the P24. And we agreed mutually after trying it out that 8 per day was the rate we were going to go for. So they were going to extract 8 and then pass that on. They were going to do the library prep with those 8. And we wanted it to be very nice and use, on the P24, you have 3 rows, right? With 8, it's going to be beautiful. That's what we set out to do. So this is one of our first samples. And as you probably know, if you've ever run a PromethION flow cell, the more green, the better. Yeah, so this is like, I was like, it worked. And I will say that this actually happened after one of our lovely technicians decided to do his own thing and added loads of bubbles into like 8 flow cells. It was painful. So I had to go in the lab and like redo it because we wanted to make sure we hit the time. But we eventually got there, which was great. And then we hit the next hurdle because the lab team was so efficient, they got into a great flow, and then we ran out of reagents. And I was like, guys, why didn't you record what you were doing? They're like, oh, we were just, happily doing all the library preps and loading them. So we did have to take a bit of a break. That's an empty freezer, basically is what that is. And so it gave us time to reflect. Everyone kind of looked at what their process was. How can we record the data so that we can actually go back and look at the process? And I will say, this is a screenshot of our spreadsheet. We tracked everything. We knew what was going on with which sample was on which flow cell. If we had to run another flow cell, we had that. I love a spreadsheet. Anyone else love a spreadsheet? Don't be ashamed. Love spreadsheets. And we also kept track of when we were washing. So we got like the pre-wash number, and then we could see like which ones actually increased. And one of the really cool things I love about this machine is you can remotely access it. So I work from home mostly, right, as CEO of the company, and I'm not myself in the lab, but I am the obviously senior scientist. So I would just log on from my bed, essentially, from home, and watch and see what's going on. And it literally became a pastime for me. Like late at night, I would just like log in and just watch the green moving around, like, it's working, and just take it in. I absolutely love that. Okay, so then we got to a point where we had everything, we were generating data, and then we hit the third hurdle, which we were generating data too fast, because the actual tower for the P24 can hold up, I think it's 55 terabytes of data, and a human genome was generating at 30x around like 1.4 was on average. And so we quickly was maxing out the PromethION. So our great tech partners worked with us. And I literally, well, first of all, we're the first in the country to generate data that fast. Our data center, and just for context, Bermuda is actually like a reinsurance and insurance hub of the world. We have the big four, we have huge names. Lloyd's London is reinsured by companies in Bermuda. So we have lots of sensitive data, and we were using that data center to store our data as well. We completely maxed them out, even to this moment. We have used all their space. But what I actually also had to do, because we didn't have like a direct line from the college to the data center, is put it on an external hard drive, put it in a very nice bag, and put it in my car and drive it. And I felt like I was, in the CIA or something. It's like really top-notch data, and literally manually deliver it. So we're hoping that we can actually set up a nice cable, so we don't have to do that anymore. But that's what we have to do in this particular study. And I want to say as well, always, always wash. Obviously, watch the run. And if the pores are below about 2,000, we're like, oh, let's not wash that. But we washed everything that was above 2,000 pores, and we kept every flow cell that was above 1,750 pores. It was a very arbitrary decision to pick that number. But what we did was we kept them. And if there was a sample that didn't quite get to the data goal from just one flow cell, we would then use a backup to supplement. And we found that was really helpful. So we have a whole system in the fridge, the backups go in this drawer, or the fresh ones go here, and it allowed us to get through everything more efficiently. Okay. Then, I feel like there's never-ending hurdles here. Then when we finished sequencing on March 19th, we were so excited. And Mitten had already taken the first batch and was analyzing them and doing the alignment. 5 days after that, the college where we were at had a hack. I don't know what's going on. But it meant we had no internet for 3 weeks. And we were monitoring the situation. We had the deadline for this. So I did have the speaker support like, hey, Dr. Weldon, we need your talk. I'm like, I'm coming, don't worry, we're trying to fix something, And so what we ended up doing was literally buying mini hard drives, putting the FASTQs on the hard drives, and shipping them by FedEx to Mitten, and you got it the next day. And then 2 days after that, they got internet back. It was like, urgh. But we had to do what you had to do. And then, while we had that short break, we actually were able to connect with GenX, and I'm not a bioinformatician, as I told you, so I didn't know how to do the command line to just upload all 102 files. So I literally got the files at 4 a.m. on April 27th, and I sat there from about 9 in the morning, and I uploaded one by one each sample, because I was like, I am getting this done. And I did that for took a bit of a break to sleep, but by the next morning, I had finished all 102. And by noon, so about 36 hours after that, I was able to do the analysis on all the samples on a weekend, right? So that was huge because we were going to have to do it manually, and that meant more time, and I wouldn't have a talk to give you here. So super shout out to GenX for that. And when it came down to picking up the SNPs, we picked up all of the SNPs that we sent off for clinical confirmation. Because we are a research lab and we were going to report back results to clinical patients, we had to get it confirmed at the clinical lab. And we picked up everything they got, but we found more. So one of the interesting things is in MSH3, they were seeing something going on in a repeat area. And we're like, we can't tell, there's something there, but we don't know. So what we did was we got the BAM files and opened up IGV. And you can see very clearly it's those top 3, where there is an insertion, is about 26 base pairs, plus about 2 base pairs over, there's a SNP. So they were the calls were getting confused because they're using short read sequencing, but we were able to see it very clearly in the data. What's also cool, and I mentioned earlier, is that we can see insights now into small island population structure. So this is also in, I think it's in MSH3 as well. There is in 96, as I mentioned earlier, of the 102, about a 300 base pair deletion. And we didn't think it was real. Initially, like, wait, what's going on? But again, looking at the data, you can see the first 3 or 4 are heterozygous, and the ones at the bottom are homozygous. It's completely gone. You don't see anything for this location. So we are now understanding, like, what does it mean, to have a small island population for the first time. Methylation analysis is underway. So I did now learn how to do the command line for Dorado. So that is an extract of what it looked like yesterday. And no extra cost, which is great. We're using the same pod 5s that we generated initially. And we're now rerunning the data, and we will get rid of those pod 5s because that's what's taking up most of the space. And again, as I mentioned before, doing the ONT whole genome sequencing, if you can imagine, is cheaper than what the insurance companies were paying for a 91 gene panel. So we now have the insurance companies asking, like, can you please do this for us? We want it local. We want to be, keeping the money in country. And so we're in the process of getting that sorted. And I want to give a huge shout out, I actually didn't know I was going to do this, to my account manager and FAST team. So Natalia, who's here, Rebecca Smith, and Sandra, who, if there's a problem, we WhatsApp them. They're like, okay, let me just get some information from the epidemic team. Or like, they are so amazing, which meant that we could get our stuff done. Thank you so much.